students, this is Suyashivanit and you are hearing me on Neetshala, #audiobook of Neetshala. So the chapter we are going to start is Sexual Reproduction in Flowering Plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swan over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescence, flowers and floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and the seeds. In this chapter, let us understand the morphology, structure and the process of sexual reproduction in flowering plants, angiosperms. 2.1 Flowers, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of the 5 more flowers that are used in social and cultural celebrations in your family. Have you ever heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In class 11th, you have read the various parts of a flower. Figure 2.1 will help you recall the parts of a typical flower. Can you name name the two parts in a flower in which the two most important units of sexual reproduction develop? So you can see here stigma, anther, petal, filament, style, sepal and ovary. It's a diagrammatic representation of lateral section of a flower. 2.2 Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated that lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In a flower, the male and female reproductive, the androecium and gynoecium, differentiate and develop. You would recollect that androecium consists of a whole of stamens representing the male reproductive organ and gynoecium represents the female reproductive organ. Okay, so let's move forward. Stamen, microsporangium and pollen grain. Just a second, let me take a marker so that we can mark properly. Okay. Figure 2.2 shows the two parts of a typical stamen, the long, slender stalk called the filament and the terminal, generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus of the petal of the flower. Sorry, I'll repeat these sentences once again. Look here carefully. Figure 2.2a shows the two parts of a typical stamen, the long slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of filament is attached to the thalamus or petal of the flower. Now you can see here in the diagram, this one is your anther and below is your, see the diagram of filament. Okay, this is the stalk like structure. Okay, The proximal end of filament is attached to the thalamus or petal of the flower. The number and length of stamens are variable in a flower of different species. If you were to collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in this size seen in nature. Careful observations of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed, you can see here, with each lobe having two theca, that is dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissue and their organization in the transverse section of an anther. Okay, so see, this is the this B diagram is the cut section view of the anther, and the first one was the typical stamen. The structure of typical stamen, which consists of anther and filament, was this figure A. Okay, so a typical angiosperm anther is bilobed. That is, each lobe have two theca. They are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissue and organization in transverse section of an anther. The bilobe nature of anther is very distinct in the transverse section of anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of the anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers. The outer three wall layers perform the function of protection and help in dehiscence of flower to release the pollen grain, and the outer wall layers consist of the epidermis, the endothecium and middle layer, and the innermost layer is tapetum. The innermost wall layer, the tapetum, it nourishes the developing pollen grain. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how many tapetal cells could be binucleate? When the anther is young, a group of compactly arranged homogeneous cells, I repeat, homogeneous cells called sporogenous tissue, occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cell of the sporogenous tissue undergo meiotic division and to form microspore tetrads. What would be the ploidy of the cells of the tetrad? Now you can see the structure here. Look here, this is epidermis. The outermost layer is your epidermis. Then you have these middle layers, and then the innermost layer is your tapetum. Here also you can see, and sporogenous tissue you can see. So these two diagrams, and this third one is a mature dehiscent anther, okay, which is releasing pollen grain. As each cell of sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell, PMC, through meiosis is called microsporogenesis. The microspore, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represents the male gametophyte. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle the grains on a drop of water and taken on a glass slide and observe under a microscope. You will be really amazed at the variety of architecture, sizes, shape, color, design seen on the pollen grains from different species. See, you can see here, it's looking so beautiful, the scanning electron micrographs of a few pollen grains in figure 2.4.